Right now, in this exact moment, somewhere inside your body, a cell is breaking its own rules. It has stopped listening to instructions. It has disabled its self-destruct code. It has begun replicating without permission. And the most terrifying part? You cannot feel it. You cannot see it. In your own immune system, the most sophisticated defense network in all of biology may have already been tricked into protecting it. Today, we are going inside a single cancer cell. We're going to watch a normal cell transform into something your body cannot stop. Not from the outside, not with statistics. From inside the nucleus, from inside the machinery. This is what cancer actually looks like. Think of every cell in your body as a factory worker in a massive city. Each worker has an exact job description. Divide when told. Stop when told. Die when told. Every instruction comes from a central rulebook, your DNA. Now, imagine one worker tears a page out of that rulebook, then another, then another. They stop reporting to management. They start multiplying uncontrollably. They consume resources meant for other workers. They build walls to hide from the security guards. And eventually they begin sending scouts to other cities, starting new rogue colonies everywhere they land. That is cancer, not a foreign invader, not a virus that entered from outside, a worker that forgot the rules and refused to die. Every cancer begins with a single event, a mutation. DNA is copied billions of times inside your body every single day. Each copy involves reading 3.2 billion base pairs, the letters of your genetic code, and reproducing them without error. Your body has extraordinary proofreading systems. Repair enzymes scan for mistakes like editors checking a manuscript, but no system is perfect. Approximately 1 in every 100,000 base pairs is copied incorrectly. Most of these errors are harmless in regions of DNA that code for nothing critical. Some are immediately detected and repaired, but some mutations land in precisely the wrong place. They hit what scientists call proto-oncogenes, genes that normally control cell growth. When a proto-oncogene is mutated, it can become an oncogene, a permanently switched on growth signal. Think of it as a car's accelerator pedal getting stuck to the floor. The cell no longer needs external permission to grow. It has overridden the brake. This single mutation is not yet cancer. It is the beginning of a conversation the cell is having with its own DNA, a conversation that, in most cases, the body interrupts and corrects. But not always. Here is the part that surprises most people. Cancer does not happen from one mutation. It requires an accumulation, typically 5 to 10 critical mutations over years, or even decades before a cell crosses the threshold into malignancy. The first mutation might disable a growth suppressor, a gene called P53, for example, which acts as the cell's own internal alarm system. P53 monitors DNA damage and orders the cell to stop dividing, repair itself, or self-destruct. Scientists call it the guardian of the genome. When P53 is mutated, which happens in more than 50% of all human cancers, the alarm system goes silent. The second mutation might activate a survival signal. Normally, cells receive continuous survival instructions from neighboring tissue. Remove those instructions, and they enter programmed death, apoptosis. Cancer cells develop mutations that generate their own survival signals. They no longer need permission to live. The third mutation might involve angiogenesis, the ability to grow new blood vessels. A tumor cannot grow beyond 1 to 2 millimeters without its own blood supply. Certain cancer cells acquire the ability to secrete VEGF, vascular endothelial growth factor, a chemical that commands nearby blood vessels to grow toward the tumor. The cancer builds its own highway system. Each new mutation stacks on the last. Each one removes another barrier. And this process can take 10, 20, or 30 years before a single cell becomes a detectable tumor. Your immune system kills cancer cells constantly. Every single day, natural killer cells and cytotoxic T cells patrol your body, identifying rogue cells and destroying them before they can multiply. This process is called immune surveillance, and it is extraordinarily effective. Most early cancer cells are eliminated before they ever form a tumor, but cancer cells that survive do so because they have evolved a way to hide. The most sophisticated mechanism is called immune checkpoint manipulation. Your immune cells carry receptor proteins, PD-1, CTLA-4, that act as molecular off switches. These exist to prevent the immune system from attacking healthy tissue. Certain cancer cells have learned to display the off signal on their surface, a protein called PD L1, essentially handing the immune cell a fake ID that says, "I am healthy tissue. Do not attack." The immune cell reads the signal, deactivates, and moves on. The cancer cell continues to divide. This is not science fiction. This is the mechanism behind some of the most advanced cancer immunotherapy drugs currently being developed, drugs designed to block the PD-1/PDL1 interaction and allow the immune system to see cancer again. But the cancer's ability to evolve means it often finds new ways to hide. It is a biological arms race playing out inside your own body. The deadliest event in cancer is not the original tumor. It is the moment cancer cells learn to travel metastasis. The spread of cancer to distant organs is responsible for approximately 90% of all cancer deaths. And the mechanism is both elegant and horrifying. To metastasize, a cancer cell must accomplish a series of steps that should be biologically impossible for a normal cell. First, it must detach from its neighbors. Healthy cells are anchored to each other by adhesion molecules, molecular Velcro. Cancer cells develop mutations that disable these anchors. Second, it must penetrate the basement membrane, the dense protein barrier separating tissue compartments. Cancer cells secrete enzymes called matrix metalloproteinases that digest this barrier, like acid through paper. Third, it must survive in the bloodstream. Normal cells detached from tissue die immediately, a process called anoikis. Cancer cells develop resistance to anoikis, floating through blood vessels and surviving circulation. Fourth, it must exit at a new location, penetrate foreign tissue, and establish a microenvironment that supports its growth in a completely different organ. The cells that successfully complete all four steps are extraordinarily capable. They have, in effect, evolved into a new kind of cell one that has taken the remarkable adaptability of life and turned it against the body that created it. Here is the biological detail that changed how science understands cancer and may ultimately be how we defeat it. Normal cells produce energy through a highly efficient process called oxidative phosphorylation, using oxygen in the mitochondria to generate large amounts of ADP from glucose. Cancer cells almost universally abandon this efficient process and switch to a far less efficient pathway called glycolysis, fermenting glucose into energy without using oxygen. This is called the Warburg effect, named after Otto Warburg, who discovered it in the 1920s. On the surface, this makes no sense. Why would cancer cells choose an energy process that produces 18 times less ADP per glucose molecule? The answer became clear decades later. While glycolysis is energy inefficient, it is building block efficient. Cancer cells need raw materials: carbon, nitrogen, the building blocks of proteins and lipids and nucleotides to build more cells. Glycolysis provides those precursors. The cancer cell is not primarily an energy machine, it is a replication machine, and glycolysis is optimized for replication. This is why PET scans, which detect areas of the body consuming unusually high amounts of glucose, can identify cancer. You are seeing the Warburg effect in action. The cancer cell's insatiable hunger for glucose, burning through fuel 200 times faster than normal tissue, lights up the scanner like a beacon. Here is the most dangerous oversimplification in popular cancer science. In 2015, a widely misrepresented study suggested that a majority of cancers were caused by random mutations in stem cells, essentially bad luck. The headline version of this claim removed personal agency and environmental context from cancer in a way that was deeply misleading. The science tells a more complete story. Yes, random replication errors occur, but the rate at which mutations accumulate and escape correction is profoundly influenced by factors you control and factors your environment imposes. Tobacco smoke contains over 60 known carcinogens, chemicals that directly damage DNA or impair the repair machinery. Chronic inflammation from obesity, processed food. Alcohol, environmental toxins creates a microenvironment that accelerates mutation accumulation. UV radiation from sun exposure directly causes the specific DNA damage seen in skin cancer. The BRCA1 and BRCA2 mutations, inherited genes that dramatically increase breast and ovarian cancer risk, are real. But even carriers of these mutations do not develop cancer inevitably. The tumor microenvironment, the immune system status, the inflammatory state of the body, all of these modulate risk dramatically. Cancer is not purely luck. It is a complex interaction between your genome, your epigenome, your immune system, your environment, and time. And that means the interventions are real. Step 1. Reduce the rate at which your DNA accumulates damaging errors. The most evidence-based interventions are not supplements or superfoods. They are the elimination of known carcinogens. Tobacco, processed and charred meat, chronic alcohol consumption, unprotected UV exposure. These are not contested. They directly accelerate the mutation accumulation process we just described. Alongside elimination, oxidative stress, which damages DNA bases directly, is reduced by consistent exercise, adequate sleep, and a diet rich in whole plant foods that donate antioxidant capacity to your cells. Not juice cleanses, not supplements. Real food, sleep, and physical activity. Step 2. Optimize the immune system's ability to detect and kill early cancer cells. Chronic stress elevates cortisol, which suppresses natural killer cell activity, the very cells responsible for eliminating cancer cells on a daily basis. Sleep deprivation has the same effect. A single night of 4-hour sleep reduces natural killer cell activity by 70%, according to research from UC Berkeley. This is not a minor lifestyle suggestion. This is measurable immune function. Regular moderate exercise increases NK cell circulation. Fasting and caloric restriction have been shown in preclinical and early human studies to upregulate autophagy, the cellular housekeeping process that clears damaged cellular components before they accumulate mutations. The immune system is your first and most powerful cancer defense. Treat it accordingly. Step 3. Do not give cancer a friendly environment to grow in. Tumors do not grow in isolation. They actively reshape the tissue around them, creating what is called the tumor microenvironment, a soup of inflammatory signals, suppressed immune cells, and recruited blood vessels. Chronic inflammation driven by visceral fat, processed food, alcohol, and dysbiotic gut bacteria creates exactly this environment, systemically reducing visceral fat, maintaining insulin sensitivity, and eliminating chronic low-grade inflammation directly narrows the window in which cancer cells can establish and grow. This is not alternative medicine. These mechanisms are described in peer-reviewed literature from MD Anderson, Harvard, and Memorial Sloan Kettering. The biology is established. The implementation is yours. Cancer is not a mystery. It is biology, and biology, at its core, is understandable. The cell that breaks the rules was once a normal cell doing exactly what it was supposed to do. The same rules that allow life to exist, to replicate, to adapt, to survive are the same rules that cancer exploits. Understanding those rules doesn't make cancer less frightening, but it does make it less arbitrary. It makes your choices matter. If this gave you a clearer picture of what's actually happening inside a cancer cell, share it. This is information that belongs to everyone. Drop a comment below. What question about cancer has always stayed with you that science hasn't fully answered yet? I want to know, and I'll see you in the next one.